All right, folks, since we're at, about a minute from our first talk, I wanted to welcome everyone to this session. My name is April Wright. I'm one of the core organizers of the Evolution meeting. We're also joined by Daniel Stanton, who is a volunteer who is here to help us with any sort of, technical issues that you might have or run into while we're doing this session today. We've got a great roster of talks lined up. And I want to remind everyone that as an evolution conference space, we are covered by the Evolution Code of Conduct. So we want to keep things positive and constructive as we're going through our day to day. So without further ado, I will introduce our first speaker, Catherine Sherd. I don't really intend to introduce future speakers, but I will introduce her since she is the first. And take it away, Catherine.
Step one, unmute. Good call. Yes. Okay. Let me share my screen. Okay. Hopefully you can all see that. Great. Thumbs up from April. Right. So my name is Catherine Sherd. I'm currently a postdoc at the University of Bristol, and today I'm going to be talking about birds that put man-made materials in their nests. And PowerPoint advances. There we go. Okay. So in 1933, a gentleman by the name of Ernest Warren wrote a letter to Nature where he said that he had observed this species, a pied crow, putting metal pieces of wire into their nest. And this was the first record in Western science of a bird putting anthropogenic materials in their nest. Since then, however, there have been a ton more records of this. Birds around the world seem to really like putting anthropogenic man-made materials in their nests. And this might be a problem, right? Particularly in the case of plastics. Birds that handle plastics will often ingest plastics, causing a disease that we now know as plasticosis, causes inflammation, often kills the birds. That's bad. There's also cases where it's not clear if it's a positive or negative effect. So birds that put cigarette butts in their nests are thought to do so because the cigarette butts kill parasites, but also the nicotine is toxic to the birds themselves. And then birds that put string in their nests, that might be good. String is very strong material, but also the string sometimes gets tangled around the chicks' necks and strangles them. But then there's cases where arguably this is evolutionarily a good benefit for the Anthropocene because it enables birds to nest in environments where they may otherwise not be able to reproduce successfully. And despite the fact that there's been lots of reports of birds incorporating anthropogenic materials into their nests, we don't have a good sense actually of which birds are doing it besides a report here and a report there. So a reasonable question one might ask is what birds are doing this? We don't know. We actually don't have that good of a sense of what materials are being put in bird nests at all, especially in the tropics of the world. Arguably in North America and Western Europe, there have been enough monitoring programs that we do have a sense for most species there of what's going in their nests. But for other parts of the world, it's not entirely clear. So I'm revising the question to what birds have we recorded using anthropogenic materials in their nest? And then once we assemble an answer to that question, we can begin to ask why. And some ideas about why some birds use anthropogenic materials and some don't are obviously going to involve a research effort, as I just alluded to. Birds that English-speaking Western science has monitored more are probably more likely to have been recorded in English-speaking Western science as using anthropogenic materials. However, above and beyond that, another fairly self-explanatory hypothesis for what might be driving this has to do with material accessibility. So birds that nest in cities or towns where there's lots of anthropogenic materials around may encounter these materials more and therefore may put them more into their nests than birds that are nesting deep into the jungle. However, on top of that, there may be a sense of underlying behavioral flexibility. Some birds are very particular about what they put in their nest. They make their nest out of only a special secret type of moss, and some birds seem to really be able to reproduce with just about anything in their nest. And maybe those birds will be more likely to introduce these new novel anthropogenic materials. It could have something to do with the type of nest, right? More complex nests with walls or maybe walls and a roof may have different costs, that the birds may be more willing and may in fact need to incorporate anthropogenic materials, or alternatively, they may be have more specific requirements for material properties, and therefore they may not be looking to incorporate new things. The breeding environment may have something to do with it. Birds nesting in the rainforest may have abundant materials available, whereas birds nesting in harsh environments may not be able to get their preferred materials and may just be making nests out of whatever they can get. And then finally, not an evolutionary hypothesis, but just something that would be good to know is, does this correlate at all with conservation status? If it's the endangered birds of the world that are the ones doing putting anthropogenic materials in their nest, that would be important to know. Alternatively, if it's the ones that are thriving in the year 2023 that are incorporating anthropogenic materials, and it's the ones that are endangered that are the really particular, only the special bit of moss birds, that would be also be very important to know. So as you can imagine, with an introduction like that, I'm gearing up to tell you about a study that has investigated these questions. So a team and I went through and compiled lists of nest materials for the birds of the world based on, again, English-speaking Western sources, primarily from the handbook, The Birds of the World, Birds in North America, Neotropical Birds Online. And then we scored these for the presence and absence of anthropogenic materials. And if you are thinking, hey, what else did you do with that? Coming soon to a paper near you, we have done other things with this data set. Talk to me again on July 10th. So we were able to get data on the presence or absence of anthropogenic material for just over 6,000 species of birds. And then also we separately scored the presence of plastic material, right? It's this non-biodegrading substance, toxic to birds, very important at the moment. So we figured we would analyze that separately. We controlled for research effort by looking at the number of papers published in Web of Science about that species. We looked at the number of recorded nest materials to try to differentiate between the ones that need their special moss and the ones that can put just about anything in their nest. We then averaged for each species their breeding range human footprint index. So this is a measure of human landscape modification all around the world. So things like cities and towns are scored very high, and things like jungles scored very low. Conservation status, straight from the IUCN. Looking at biome as a way of differentiating different environments for their breeding range, scored their nest structure, and then also scored their nest location, so where they're putting that nest, to try to tease apart these different possible costs and benefits to different nesting strategies. And in particular, we are looking at birds that have been recorded as nesting in an artificial location. So that's things like, have they been known to nest on roofs or on or in telephone poles, with the idea that that might be a measure of how well they're able to tolerate humans. You map these variables around the world. So on the top is the map of anthropogenic nest material, that's in pink. And on the bottom, that's plastic nest material specifically, that's in green. Darker colors mean more birds breeding in that region are known to use that type of material. First thing that jumps out at you is North America, which it's got to be research effort, right? So hold that thought. But if you then look at the other pockets of dark colors, you can see that there's been a lot of recordings of these materials in islands and also in the world's deserts. The Sahara and some of the Arabian Peninsula is missing because we didn't have a lot of birds breeding there in our data set. But then you can see around the edges, there's this sort of ring of dark colors. It seems that birds breeding in some of the world's major deserts are the ones that are using this. Okay, so that's spatially, but that's actually analyzed it. So we chucked everything into a big, giant phylogenetic generalized linear mixed model using McGlim. You get a giant table like this. Don't panic, I'll walk you through it. And this is specifically for the use of anthropogenic materials. I'll show plastics later. So the first thing to note from a table like this is that there's no correlation with conservation. Highly endangered birds are not more or less likely to incorporate anthropogenic material. So good to know. The next thing to note has to do with nest structure, which apparently has no correlation with the use of anthropogenic nest material. So no matter what structure birds are using, they're not more or less likely to use man-made material in their nests. However, what it does correlate strongly with are these variables that are related to human tolerance. So birds with a high breeding range human footprint index, so are breeding in ranges where there's been lots of human modification, are much more likely to use anthropogenic materials, and also birds that have been recorded as nesting in an artificial location. So that's the telephone poles on roofs or so forth, much more likely to use anthropogenic materials. Also seems to be related to breeding range biome. This is a bit hard to tell from this map, but in general, indeed, the birds that nest in deserts and also weirdly enough in mangroves are more likely to use anthropogenic nest material, and the ones that nest in tropical forests are less likely. And then finally, we have the number of materials. So these birds that have very long lists of materials that have been recorded in their nests are more likely to use anthropogenic materials than the ones that only nest using one very special type of material. And this is even after accounting for research effort and for all the other things that you see here. Cool. The model for plastics is actually very similar. The one difference has to do with the nest structure. So birds that create a scrape for their nest, or just like use a bit of lining but don't really construct a nest as such, are more likely to incorporate plastic either in that scrape or in that lining. We interpret that as being mostly driven by the seabirds. Seabirds do a lot of scrape nesting. Seabirds are highly monitored, and seabirds are also really into interacting with plastic, which is a major conservation concern at the moment. So again, this is just on the birds where we know what sort of nest materials they're using, which means that we're undercounting a lot of tropical birds. This is also, again, biased by English-speaking Western science, and the Handbook of the Birds of the World is not perfect, but we at least have a start, right? We can take a bird around the world, and using basic information about its ecology and environment, we can predict how likely it is to incorporate anthropogenic materials, which can then inform conservation decisions about this species. So with that, I am under time. Good job, me. I'll thank my co-authors and my funders. I will leave you with a nice conclusion slide. I will gladly take any questions either here or on social media. Thank you.
Wonderful. Thank you, Catherine. And we do have time for questions. So if anybody has questions, feel free to put them in, either raise your hand or put them in the chat, and I will unmute you so that you can ask that question directly if you feel comfortable with that. If you don't feel comfortable with that, you would rather just type out your question, that is okay too. Sorry, it's my kids.
Silence. Too early in the morning for those of you in the US.
It's not that early, unless you're on the West Coast, in which case probably dragging a little bit.
Yeah, East Coast is almost 11:30, so people should have woken up by now.
All right, and we've got a question from Kyle, so I'm going to ask him to unmute. All right, Kyle, you should be able to ask your question now.
Awesome. Well, thank you. Great talk. I'm just a little curious, and maybe I missed this. The model you used, you say it was like a linear, and it included phylogenetic information as well. Could you talk a little bit more about that?
Yeah, so it's using the program McGlim. So phylogeny is included as a random effect, in this case using the global bird tree from Yates et al. 2012. And there is a phylogenetic signal of both this as the response variable and for some of the predictor variables we included. So we integrated that over 100 phylogenies drawn from the posterior distribution to try to also account for phylogenetic uncertainty.
That's MCMCGLM.
Yes, sorry. Yes, MCMCGLMM.
Awesome, thank you.
Which is good if you want to incorporate over phylogenetic uncertainty. If you just have one tree, there are much faster ways to do it.
All right, excellent. So we're going to go
Thank you.
ahead and have our next speaker go ahead and get their slideshow up. Perfect. That looks.
Can you see? Can you hear me properly? Okay,
Okay.
good. Okay, great. So I am Hélène Leroy, a PhD student at Charles University in Prague in Czech Republic, and I'm doing a project on studying trait dynamics in passerine evolutionary radiation under the supervision of Vladimir Remesh. And today I will tell you about our study on patterns of trait evolution in the radiation of songbirds. So first, let's talk generally about evolutionary radiation, which are thought to be at the origin of a significant part of the biodiversity on Earth, and they are broadly defined as an increase in the diversity of a clade within a short time span. And within evolutionary radiation, adaptive radiation received the most attention, and here the increase in diversification is coupled with adaptive disparification, which allow the utilization niche. However, there is no, for instance, non-adaptive radiation, which is a case with where the increase in diversity is coupled with minimal ecological divergence. And it's been argued that some evolutionary radiation were actually prematurely diagnosed as adaptive radiation. So let's speak about a specific case, which are Parilidae or New World wood warbler, which are often presented as an exemplary adaptive radiation. They are a wide radiation of New World songbirds, and they represent more than 100 species. And indeed, they underwent a rapid diversification from a common ancestor, followed by a diversification slowdown, which is thought to evidence ecological niches. They are also a classical example of foraging niche divergence. However, they present some aspects which could be in contradiction with the definition of adaptive radiation, as for instance, they inhabit similar habitats, which are mostly woodland and forest. They also are morphologically quite uniform, which could be in contradiction with disparification to exploit different niches. And they also present plumage diversity, which is a characteristic which could have helped them to facilitate coexistence, because they present some highly syntopic species. So the goal of our study was to unravel patterns of trait evolution in the wood warbler radiation by testing alternative macroevolutionary scenario for the disparification of phenotypic traits. So I will introduce you to the main model or scenarios of evolution which we tested through traitgram or phenogram, in which you have trait value versus clade age, relative subclade disparity through time, which allow to visualize the evolution of disparity in comparison with a null Brownian motion model, which is depicted here by a gray envelope, and phenotypic rates through time. So the first model is Brownian motion. It's a model where the trait is evolving randomly across direction and distance over any time interval. And in that case, the trait changes are accumulating linearly with time. The second model, Orsten-Uhlenbeck, is a model where there is an optimum trait value toward which the population is experiencing attraction. And in that case, the traits are evolving under stabilizing selection. And if you look at the disparity, at the end, there is slightly more within clade disparity than among clade disparity. The third model, early burst, is one idea of adaptive radiation in which you will have bursts of phenotypic evolution early in the history of the clade, which is associated with the colonization of new ecological niches. And then you will have a slowdown in time as the ecological niches are filled. And in that case, you will have way more among clade disparity. And lastly, the late burst model is a diversity-dependent model where evolutionary rates are increasing with the accumulation of diversity in the clade. So you will have the phenotypic rate, which will accelerate toward time, and then you will have way more within clade disparity than among clade disparity. So let's see what we found for the wood warbler radiation. So we fitted this different model of evolution to morphological, song, and plumage traits across the phylogeny. So first, for morphology, here you have the weight of each model for all of the morphological traits. And as you can see, this is the OU or Orsten-Uhlenbeck model, which was the best fitted model for all morphological traits. And the disparity through time was evolving according to a Brownian motion model. So we have a gradual phenotypic evolution with an attraction towards an optimal trait value, which is in accordance with the morphological similarity which we find in this bird. And this could be because they specialize in ecologically similar environments. We found similar patterns of evolution for song traits. So the Orsten-Uhlenbeck was again the most fitted model. And for most traits, the disparity was evolving according to a Brownian motion again. So it is possible that the evolution of song trait was constrained by morphology because it has been found, already that big morphology and body size diversification was driving patterns of vocal signal evolution within birds. And this could also be due to an adaptation to enhance signal transmission in shared habitats within coexisting species. And for plumage, the result we found stood out in comparison with morphology and song. So we found characteristic typical of a diversity-dependent burst of trait evolution, which happened late in the history of the clade. So the most fitted model was a diversity-dependent exponential model. So the evolutionary rates exponentially increased with the number of species. So we have, and you can see here that the disparity through time were increasing for within clade disparities. A potential explanation for this is that evolutionary rates of color traits accelerated with secondary contact, with species interaction promoting color signal divergence to reinforce species recognition. And as there is little ecological divergence, this could have facilitated local coexistence. So to summarize, the scenario of the radiation of New World wood warbler does not completely fit a scenario of adaptive radiation. So yes, we have a rapid diversification followed by a slowdown, but it seems that this diversification happened in allopatry in ecologically similar environment, which resulted in minimal ecological divergence. And after a period in allopatry, they either stayed allopatric or became sympatric due to a modification of their ranges. And then the species coexistence was most likely facilitated by behavioral niche partitioning and signal divergence, which most likely evolved after secondary sympatry. So they present signs from both adaptive and non-adaptive radiation. So we think that they are part of a continuum between non-adaptive and adaptive radiation, and that this could be the case for many evolutionary radiation. And thank you for listening.
Excellent. Well, we have lots of time for questions. Very lovely talk. So if anyone has questions, again, you can raise your hand and I will ask you to unmute so that you can ask your question in person. If you're not comfortable doing that, you can certainly put your question in the chat to be answered.
Okay, so no question, I guess.
Yeah, people are always a little shy towards the start of meetings. Oh, wonderful. I've got a question from Orlando.
Hello.
Perfect.
Hello.
We can hear you.
Yeah, very nice talk.
Thank you.
Just a very brief question. So you mentioned that rapid diversification and then a slowdown would hint towards adaptive radiation. I was kind of wondering what your reasoning is why that couldn't also be observed in a non-adaptive one.
You mean the rapid diversification or the slowdown? But I can hear you. I cannot hear you. Okay.
So I've learned that if you mute yourself, you can't unmute yourself again.
Okay.
So we should keep that in mind. Yeah, so I guess you presented both of these, like the rapid radiation and the slowdown, as like hinting towards being an adaptive radiation.
Yeah, sorry. So I don't necessarily think that the rapid diversification is a proof for adaptive radiation, and the slowdown, however, it has often been presented as an evidence that the new ecological niches which appeared, which generated the adaptive radiation, are a proof that these ecological niches are filled. But however, there is some discussion around that, and if the paralids are not an adaptive radiation, then there must be another reason for this slowdown, which could be many reasons.
All right, thanks.
Thank you.
We have one more question in the chat from Catherine. Do you think the paralids are especially different from other songbird clades, or do you think this result will generalize?
That's a great question. I actually don't know, because you have to study other radiation, and I'm not necessarily initially a bird person, so I'm not sure, but that's an interesting question. Like, I will later on study another family, so I will have other results. That would be interesting.
All right, well, if there are no other questions, thank you very much for answering those, and we can start our switch over to our next speaker. Nicholas, if you would like to share your screen now so we can get that going. Perfect, we can see that. And I'll give you a heads up right at 10:45 or whatever your local time is, 15 till, so you can get started on time. All right, you are a go.
All right, well, good morning, everyone. I'm Nick Levis, and I'm a postdoc at Indiana University working with Eric Ragsdale. And today I'd like to discuss the mechanistic basis for developmental plasticity's role in the evolution of novelty and diversity. And so developmental or phenotypic plasticity can be defined as the ability of an individual organism to alter some aspect of its phenotype in direct response to changes in its environment. So to illustrate this, here's some examples. This horned ghost crab has different coloration depending on the time of day. This common water crowfoot has different leaf morphologies depending on whether or not the leaves develop above or below the waterline. And this worker and super soldier seed harvester ants exhibit these different phenotypes based on early life nutrition and growth. So as you can see, the differences brought about this within generation plasticity can be as great or greater than some of the differences we see between different species. And more generally, plasticity has been linked to macroevolutionary changes like the origins of novelty and diversification. And it's exploring the mechanistic basis of this link that I'll be focusing on my talk today. And I'll be doing this in the context of diplogastrid nematodes that exhibit a resource polyphenism, which is an extreme form of phenotypic plasticity characterized by distinct resource use morphs. So on the left here, you can see this FT, which we call the stenostomatous form. It's a microbivore. It's got a narrow mouth and a spade-like dorsal tooth. This morph particularly predominates under low density conditions when there's lots of bacterial food, and it develops relatively quickly. However, if conditions worsen, there's bacterial foods running out, the worms are at high density, some individuals may switch their developmental program and develop into what we call the eurystomatous, the EU, this wide mouth form, where that tooth develops a claw-like shape and it develops an opposing subventral tooth. And so this morph does well under these conditions because in addition to eating bacteria, it can also predate on other nematodes. So neither form is intrinsically better, but their fitness and cost benefits of being one or the other depends on the environmental context in which they develop. So in the family Diplogastridae, this resource polyphenism is ancestral to the entire group. So if you've heard of C. elegans, that's not part of our group, that's an outgroup here. However, if you look at this tree, there's lots of blue and red across it. And this red indicates the loss of plasticity. And this is called genetic assimilation, and it's happened multiple times independently across this family. And in this case, genetic assimilation is associated with increase in the rates of mouthform evolution. So to illustrate this point, if you look at this large red clade where plasticity has been lost, they have some of the highest rates of mouthform evolution. So they're undergoing the most changes over time in the way their mouths look. And so genetic assimilation generally is not well understood at the mechanistic level how it occurs, but this situation is perfect for trying to understand generalizability of what genes are targeted during the assimilation process and to start to understand the mechanistic basis for this pattern that we're seeing here with increased rates of evolution following assimilation. And we hypothesized that genes whose expression was previously polyphenism biased might bear some signature of evolution following the loss of plasticity. So as a first step towards understanding this problem, we sought to perturb or ablate switch genes that regulate the polyphenism to understand the genetic targets of the polyphenism. This has been done previously in the model organism Pristyanchus pacificus using a combination of forward and reverse genetic techniques. And as proof of principle that these mechanisms could be targeted and manipulated in non-model species, I looked down at this species here, Pristyanchus physidentatus, and use CRISPR-Cas9 to knock out these switch genes as well and infer its polyphenism targets. And by comparing these two groups, we can there identify 4 set of polyphenism genes that are likely ancestral to this entire group. And so that's what I did. I used RNA sequencing and CRISPR-Cas9 to identify targets in physidentatus, compared them to targets in Pristyanchus pacificus, and identified a set of about 800 concordant differentially expressed or differentially polyphenism associated genes. And with this set of genes in hand, I could then look at their evolution in these assimilated species to see if there's anything shared across independent events. So here's that tree again. And for this analysis, I used existing transcriptomes and genomes that have been published. And one scale, I looked at 2 species that have undergone assimilation within the genus Pristyanchus. There's 41 transcriptomes for 41 species across this family. And I compared the results of that analysis to one using genomes spread across the entire diplogastrid tree, including 2 assimilated species here, as well as outgroups in various polyphenic taxa. So I was looking for genes that exhibited an increase in evolutionary change at both of these events, and focus especially on those genes that are polyphenism associated. And I came up with a list of 5 candidates. Looking at these and their putative functions, I chose to functionally validate this gene SPUR5. SPUR5 is a histone demethylase, meaning it removes methyl marks from histone proteins and thereby can regulate transcription of various genes. In the model organism C. elegans, it's been shown to be a regulator of transgenerational epigenetic inheritance. So histone marks accumulate and can be passed from generation to generation for things like fecundity and longevity, at least in examples in C. elegans. More importantly for me, there's in the literature a theoretical and conceptual link between this non-genetic or this transgenerational inheritance and genetic assimilation, such that epigenetically modified or transmitted phenotypes might form a sort of bridge that buys time for genetic fixation of a previously induced trait to take hold. And so I wanted to explore the effects that SPUR5 perturbation has on a polyphenic species to learn more about its potential role in genetic assimilation. So again, that loss of plasticity. So first thing I did was engineer a mutant with a loss of function allele and inserted it into our model polyphenic species, Pristyanchus pacificus, and then reared worms in 2 environments, one that favors the ST, that narrow mouth form, and one that favors the wide mouth EU form. And so what I'm showing you here is the proportion of worms developing as the wide mouth form and our wild type. Almost all of them are the narrow mouth in this environment. Almost all of them are the wide mouth in this environment. And that plasticity is greatly reduced in the mutant background, where we see a massive drop, especially in the NGM, the EU inducing environment. And as we were looking at these worms under this microscope, we noticed some of them had relatively intermediate morphologies that are almost always absent in wild type strains. And so whether we bin these intermediates as being EU or ST, there's still a significant reduction in EU. But here's an image of what some of these might look like. So here's one intermediate, its mouth is narrower than a typical EU. The aspect ratio is different, but it still has this claw-like tooth and on the dorsal side of the mouth. So rather than relying on my eyes to say that's intermediate, we can quantitatively assess this using geometric morphometrics, putting landmarks and sliding landmarks around the mouth to characterize the shape of wild type isolates as well as mutant worms. So we collected wild isolates of Eurystomatus, Stenostomatus, as well as my mutants, plotted them in morphospace, and looked at how far apart they were and phenotypically. So each point here is an individual worm. The symbols are different genotypes. What I want you to focus on are the red and the blue. So these are wild type ST and wild type EU. They're completely non-overlapping. They occupy different regions of morphospace. But then if you look at the purple and the orange, these are my mutant ST-like and EU-like as determined by a model-based clustering approach. You'll notice that they are overlapping in their ellipses, as well as bleeding into this intermediate space that was unoccupied by the wild type forms. So what all of this is showing us is that SPUR5 modification influences plasticity as well as the phenotypes that plasticity develops, and is causing phenotypes that were previously unoccupied in morphospace to arise. So seeing that it influences plasticity, I wanted to determine whether modification of this gene is influencing the polyphenism switch, or if it also affects downstream genes, downstream of the switch. The reason being that we know that after genetic assimilation or associated with assimilation in this family, there's been this diversification of forms. So we wanted to see if SPUR5 still affects mouth morphology, even after, or even if we fix what phenotype is produced. So for this, we used a line that's engineered to produce only the Eurystomatus form. So 100% of the worms are Eurystomatus. I crossed in my SPUR5 mutation and then used geometric morphometrics to evaluate whether the double mutants look different from the single mutants. And indeed they did. The gold worms were significantly different in morphospace, multidimensional space, than the purple worms in this case. So we're seeing that SPUR5 might be playing a role in the reduction of plasticity, the emergence of new morphologies, and further diversification of those morphologies. Finally, because this gene is a histone demethylase, it can result in accumulation of epimutations. So in a homozygous genetic background, modification of this gene can result in accumulation of methyl marks throughout the genome that might be selectable substrate for driving shifts in mouth form ratio over generations. So I want to note that when people have tried to select on mouth form in homozygous lines, wild type lines, they were unable to drive shifts in the mouth form ratio because these worms are hermaphroditic, they're clonal, so there's very little genetic variation. We surmise modifying this gene would allow epigenetic or non-genetic variation that maybe we could select on. So I established 3 lines, one where I selected for the EU morphology, one where I selected against it, and one where I've quantified the phenotypes, but randomly chose which plate to use to propagate. At the beginning, the lines didn't differ from each other in morphology, but over 10 generations, they significantly diverged in their EU to non-EU ratio. I then randomly sampled worms from each of these populations and did geometric morphometrics on them and found that they do indeed occupy different regions of morphospace. So if you look at this purple group, there's an ST-like and an EU-like cluster. The red, where I was selecting for the EU, is shifted much more towards the EU side of the spectrum. And then the blue actually has some ST, but then has widened its diversity, consistent with the idea of character release or fewer constraints, because it's no longer trying to express a particular phenotype. So this suggests that we can select on these epimutations to drive shifts in the mouth form and the mouth phenotype as well. And so to sum up what I've gone through here, I used comparative functional genetics to reveal common molecular targets of polyphenism across the genus Pristyanchus, and then evaluated the evolutionary history of these genes under 2 independent genetic assimilation events, and found that the histone demethylase SPUR5 has diversified following multiple losses of polyphenism, that this gene mediates the plastic response and morphologies wrought by plasticity, and that it can drive transgenerational shifts in mouth morphology. Therefore, modified SPUR5 function may provide an epigenetic bridge to genetically heritable change that canalizes a previously inducible phenotype. And so this work gives a mechanistic face to accumulating theory, suggesting the importance of epigenetics and non-genetic inheritance in plasticity evolution and genetic assimilation. And so with that, I'd like to thank the Ragsdale Lab for their support and discussions about this work, and all of you for watching my talk. Thank you.
Excellent. We do have time for questions. So if anybody has a question for Nick, you are welcome to raise your hand, and I will extend you the permission to unmute. If you're not comfortable doing that, you are welcome to put your question in the chat where it will be answered.
Hey, Nick, actually, I'll ask a question. Great talk. You talk about plasticity and methylation, but thinking about worms and thinking about how their mouth shape is different, is this driven by food source or environment, or, you know, if they eat different types of small, I don't know what these worms eat, but I work with worms as well, paramecium or whatever. Does food source matter in the type of mouth you see?
Yeah, as far as we can tell, it does matter. That's a great question. In the lab, we rear them on a pretty standard diet of bacteria, but other labs have done a wide sampling, and you can shift the mouth form ratios. Whether it's the nutritional value of the food or the ease of access is unclear. There's some thinking that those teeth might be used for popping fungal spores, as well as being used to break the cuticle of other worms. So there is definitely this environmental component And how much of it is early life diet versus like pheromone and environmental sensing isn't clear yet. So it's like a metabolism driven thing, or is it, oh, the environment's getting crummy and I'm running out of food kind of thing? We don't know that level of the cue detection yet.
Yeah, I know this is a bad analogy, but it's kind of like cats in a box, right? You know, if a cat fits, a cat sits. So I was thinking like with mouth, if the worm only has a certain size of food available, that it may adapt in a way so that it can still not go hungry. And so that's what's kind of curious about, if like food size may be a driver of mouth evolution.
Yeah, so I mean, that's certainly a good idea. And then these worms may have evolved the polyphenism to cope with situations where easily accessible bacterial food isn't available. So they're running out of that. Well, there could be this, there's other evidence out there, this is co-option of other networks. And now some individuals are able to develop this wide mouth to take advantage of other food sources. That's how the polyphenism might have started out. And so we know that they can switch back and forth depending on the environment, but what the precise reason isn't as clear. And yeah, other questions, please email me, because it seems like we're ready to move on.
Yep, we are ready to move on to our next talk. And you do have one question in the chat if you
Maybe
wanted to answer that there.
I can answer it in the chat there, yeah.
All right, Yimin, you ready to go?
I have to unmute. Yeah, I'm ready to go. Can you hear me?
Looks good. You're a go.
All right. Thank you. Hello, everyone. My name is Yimin. I'm a postdoc in University of Florida, and today I would like to introduce some of my work. I feel that's some quite of interesting finding, but those are pretty much preliminary. But I do want to share some of this because it looks very cool to me. It is. Well, so I would like to start from. Sorry, I kind of forget this. So I would like to talk about the evolutionary history of Lepidoptera along with the angiosperm radiation, because we already know a lot of moth and butterflies, they're diversified with angiosperm species, but we actually haven't ever really looked at like genomic level evolution to see if we can find any evidence supported from the genomes. So this is what I would like to talk about. And I would like to start with the most recent phylogenetic tree, which reveals the evolution of Lepidoptera and its diversification along with the angiosperm radiation. So there are about 37 superfamilies in this order, and you can, and it was clustered into 24 lineages based on the phylogeny you can see on the screen. And I'm not going to use those scientific name because there are a lot. I'll just use the number so you can see I code those clusters into 1, 2, 3, 4, 5, all the way to 24. Those are the evolutionary lineages I would like to use in this presentation. But there is another simplified version of the tree so that I can kind of outline the evolutionary history of Lepidoptera and its sister order Trichoptera. So Trichoptera, usually we refer to the caddisfly here on the tree, is a sister aquatic aquatic order of Lepidoptera, which is moth and butterfly. And if you move on along the evolutionary path, you start seeing the lower lap or primitive plate of Lepidoptera. Here I put microturgidity. And we continue to move on, and we start seeing the proboscis, which is a very unique structure to a lot of butterflies and moths. As an adult, they can feed on the nectars of flower, and this is only start from the lineage number 5. Here it's Adiloidea, and then we continue to move on, and we start seeing a new morphological innovation called Ditresia. Here start from the lineage number 7 or Tinoidea. They have 2 distinct sexual openings, one for copulation and one for laying eggs. So Ditresia is also the most diverse group, which account for about 98% of the species in this order. So you can imagine everything green here are very diverse compared to Glossata, which is in blue, and or even other lower Lepidopteran lineages. And interestingly, this time since matched to the angiosperm radiation in Cretaceous, we believe there are some connections between them. I mean, the radiation of Diatricia and the radiation of angiosperm. So we're trying to find out some interesting result. But the first thing I feel odd is like, for those non-Diatrician lineages, like between lineage 1 and before lineage number 7, they are actually also feeding on angiosperm, even though when they evolve, there are not many angiosperm species on the Earth. So I wonder maybe they have adapted secondarily to angiosperm, just like there's cousin of Diatrician species. But if putting together with the fact that the Diatrician species are much more diverse compared to those non-Diatrician lineages, as you can see here on the y-axis, here is the superfamilies which are clustered into Diatrician and non-Diatrician lineages. You can see on the x-axis here, number of species estimated from the literature. Diatricia has way more species than non-Diatrician, even though they both are feeding on angiosperm. So the question becomes why the non-Diatrician species didn't radiate as their cousin Diatrician lineages. So I would like to look at the genome to see if we can find any clues. So I started data mining. So from Darwin Tree of Life and NCBI, I collected about 40 genomes of Lepidoptera and then our group 2 caddisfly genomes. And those are high quality. How do we know this? Because we use BUSCO to evaluate the completeness of the genome assembly. So the idea of BUSCO is to use a set of genes that are putatively conserved and remain single copy in most of the species. So for example, the Endopterygoda gene set has been, which has about 2,000 genes. They're mostly single copy genes to the insects they undergo the pupa stage when they have metamorphosis, such as beetles, wasps, bees, moths, caddisfly. And because I use those high quality genomes from the database, so of course they are high BUSCO completeness across between 95% to 100%, including the 2 caddisfly species here on the far left here on the graph. You can see they have BUSCO completeness about 96. But if we use the Lepidoptera gene set, which has about 5,286 genes, we should see the BUSCO score about the same for those Lepidoptera species, but should be lower for the caddisfly because caddisfly is not Lepidoptera, right? So they may have missing some of the genes. But if you look at the BUSCO database constructed by the 37 species, they're actually from more recent derived clade. So I don't know whether we should expect to see the low BUSCO value for those unsampled lineage on the left in the non-Diatrician lineages. So here's what we found. We found that the BUSCO score for those primitive lineages are quite low, almost comparable to those like caddisflies. I believe this is something like a biology or universal to those low lab, because we have several samples from those lower lab lineages, and they are sharing the same missing gene sets from the BUSCO gene database. So this is something that we would like to know about, like if we investigate the missing genes or the gene that new to the more recent derived clades, we would probably like to see some genes that show some very important ecological innovation. So before going that far, I would like to introduce some of the most phenomenal ecological and morphological innovation along the evolutionary path of this animal. So first, from the caddisfly to the first moth, they switch from aquatic to terrestrial habitats and then start feeding on plants. Although it's mostly in that stage, it's mostly liverwort and moss, non-vesicular plants. and the remarkable morphological innovations from the hair-like structure to the scales. So in caddisfly, they don't have scales on their body or wings. They only have those hair-like structure. Excuse me. For the adult feeding, and once we start going to Lepidoptera, we see another important new innovation about adult feeding behavior with proboscis. That it's the name we gave it for Glossata. So that starts from lineage 5, and then most of the Glossata species feed on vesicular plants, and that's what we know about that group. And then we continue to move on, and we gets into a subclade called Ditricia, which again, it accounts for 98% of the species. It's defined by the two distinct sexual openings, one for occupation, one is for mating. So one's for mating, one's for laying egg. And we see that Ditricia has more genes getting. And another important thing for this lineage is most of the lineages in Ditrician species, the Ditrician lineages, they have switch from leaf miner to external feeder, so they can get very large caterpillar, and probably that's why they get extremely diverse. That's still a question mark there, but we just want to know what exactly they're about their missing gene, and can we relate it to those ecological and morphological innovation. So here's the enriched function from those missing genes, Busco genes. So from caddisfly to the first clade of Lepidoptera, we see a lot of functions enriched about the neuron cell morphology, including microtubule and cytoskeleton. From the first Lepidoptera lineage to Glossata, and we start seeing genes involved in sensory receptors and digestions. We believe that this implies that the adaptation to the host plane were very important in this step. And then similar genes, we continue to see the similar patterns. Many of them can be clearly linked to their ecological innovation, but there are some problems here. What we have done this is based on just single copy genes from Busco data sets, which is only a small portion of the entire genome. And then the other problem is these genes, because they're missing in those clades, so we won't be able to annotate it. We have to use the other genomes with the same orthologs or same single copy gene annotation to obtain the function, which may cause a bias, because once you have gene duplication, which is very likely to that specific species, in this case, I use Bomis mori or silkworm, that would bias the enrichment analysis. So we started to develop the pipeline. So we started from data mining. Again, I collected about 40 genomes from Darwin Tree of Life and NCBI, and I ran Busco on those gene assemblies, and I used the single copy gene data metrics to create a phylogeny. At the same time, I start collecting the protein database and the RNA sequence for those 40 species, and trying to re-annotate those genomes to standardize annotation pipeline. The main reason doing this is just kind of a remove or suppress the noise. Then with those gene sets from the breaker pipeline gene model, we use EGNO to annotate the genome to get the function of those genes. At the same time, we get the goal term annotation with gene ontology terms. You can imagine this is just a summary of the functions of those genes. At the same time, I use AutoFinder to cluster the gene sets for those 40 gene sets to find the author groups. Here it refers to HOGs, HOGs. It's a hierarchical author groups. You can imagine those are similar to gene families or the gene that cluster, because they're similar in terms of their sequence and the function. And from those HOGs, I extract out those genes that are new to Lepidoptera, which means they're missing in caddisfly genomes, but existing in most of the Lepidoptera species. and then so forth and so on. So we have hawks new to Glossata, we have hawks new to Diatricia. And then for those hawks, we use those 40 annotations to repeatedly do enrichment analysis. And then we eventually find a consensus enrichment set or enrichment goal terms across all the 40 species annotation, and then to find a consensus so that we can avoid the bias from a single species annotation. So here's the result very quick. Surprisingly, we only find 4 new hawks from caddisfly to moth and butterfly, but a lot more are from the basal part of the Lepidoptera to Glossata and Diatricia. And we see this, the 4 hawks, a lot of them are about the body hair scales and cellular morphology. I think this is interesting because we have seen the morphological innovation from the hair-like structure to the scales, and we also seen flight behaviors. At the same time, we saw a lot of hawks unique to Glossata. Yeah.
I unfortunately have to stop you there so that our
Okay.
final speaker can go.
Yes. Sorry.
But, oh, no problem at all. Certainly, I'm not looking at the chat right now, but if there are any questions in there, feel free to take them.
Yeah, please. Thank you.
All right. And let me just stop our recording since our final participant has asked not to be recorded. So I will